Today, we're diving into a topic that's both terrifying and fascinating: where new viruses come from. You might think viruses are just tiny gremlins that exist to ruin your day, but there's a whole lot more to their origin stories. From the depths of bat caves to the sterile labs of scientists playing God, viruses have some pretty wild backstories. First off, let's get something straight. Viruses are microscopic freeloaders that can't even reproduce without hijacking a host's cells. They're like that one roommate who eats all your food and never does the dishes, except instead of dirtying up your kitchen, they're out here causing pandemics. And boy, do they love to mutate, always looking for new ways to sneak past our immune defenses. But how do these little punks actually come into existence? Well, it turns out that nature has a way of stirring the viral pot. Animals like bats and pigs are often the unwitting hosts of viruses that, given the right conditions, can leap into humans. It's like a game of viral hopscotch, except the stakes are a bit higher than skin knees. Of course, human activities make things even more interesting. We're tearing down forests, expanding cities, and basically rolling out the red carpet for new viruses to emerge. And let's not forget those times when viruses escape from laboratories, or worse, are deliberately created. Yeah, that's a thing. Introduction to viruses. Alright, first things first. Viruses are tiny, infectious agents that are about as close to being alive as your leftover pizza from last week. They can't do anything on their own. No eating, no moving, not even reproducing. For that, they need a host. So they invade the cells of living organisms, hijacking their machinery to churn out more viruses. Think of them as biological hackers. Viruses come in a variety of shapes and sizes, but they all share a basic structure: genetic material wrapped in a protein coat. This genetic material can be either DNA or RNA, and it holds the instructions for making new viruses. The protein coat protects this genetic blueprint and helps the virus latch onto and penetrate host cells. Here's where it gets interesting. Viruses are incredibly good at what they do, mainly because they evolve at an astonishing rate. RNA viruses, in particular, mutate so quickly that they can develop resistance to antiviral drugs in no time. This is why it's so hard to create long-lasting vaccines for certain viruses, like the flu. Now, you might be wondering, how do viruses actually affect the body? Well, when a virus infects a cell, it starts replicating like crazy. This can kill the cell, cause it to malfunction, or make it a factory for producing more viruses. The immune system kicks into high gear to fight off the infection, which is what causes many of the symptoms we associate with viral diseases, like fever and inflammation. But here's the kicker: viruses don't affect everyone the same way. Men and women can experience different symptoms and outcomes from the same virus. For instance, during the COVID-19 pandemic, it was observed that men were more likely to suffer severe outcomes compared to women. This difference can be attributed to a combination of genetic, hormonal, and behavioral factors. Estrogen, for example, has been found to boost immune responses, giving women an edge in fighting off infections. Natural reservoirs, the origin story. Let's dive into the wild world of natural reservoirs, the cozy homes where viruses like to hang out before making their grand debut in humans. A natural reservoir is typically an animal or insect that harbors a virus without getting sick. These reservoirs act as viral breeding grounds, letting the virus multiply and mutate until it stumbles upon the right combination to infect humans. It's like a viral internship program, except instead of gaining work experience, the virus is gaining pandemic potential. Bats are the rock stars of viral reservoirs. They host a staggering number of viruses, including some of the most notorious ones like Ebola, rabies, and coronaviruses. Bats have a unique immune system that allows them to carry these viruses without getting ill themselves. This makes them perfect viral incubators. The close quarters in bat colonies, combined with their ability to fly long distances, make them ideal for spreading viruses across regions. Let's talk zoonotic transmission, the process by which a virus jumps from animals to humans. This usually happens through direct contact with the animal, its bodily fluids, or contaminated surfaces. Sometimes, an intermediary host gets involved, like a pig or a camel, which acts as a bridge between the reservoir and humans. The Hendra virus, for example, is carried by fruit bats in Australia. When these bats drop fruit that horses eat, the virus can jump to the horses. Humans then get infected by close contact with the sick horses. Take SARS-CoV-2, the virus responsible for the COVID-19 pandemic. It's widely believed to have originated in bats, then possibly passed through another animal species before infecting humans. This zoonotic hopscotch isn't rare. About 60% of all infectious diseases in humans are zoonotic in nature. But why do these jumps happen more frequently now than in the past? Human activities play a massive role. Deforestation, urbanization, and the global wildlife trade disrupt natural habitats, bringing humans and wildlife into closer contact. This increases the chances of viruses making the leap from animals to humans. Gender dynamics come into play here too. In many cultures, men are more likely to engage in activities that put them in close contact with wildlife, such as hunting and animal husbandry. This higher exposure risk makes men more susceptible to zoonotic infections. Women, on the other hand, might be more exposed in settings like marketplaces where animals are sold, or in caregiving roles, where they tend to the sick. Virus evolution and mutation. Now, let's get into the nitty-gritty of how viruses evolve and mutate. This is where things get technical, but it's crucial to understand how these tiny terrors stay ahead of our defenses. Viruses are like the ultimate shapeshifters. Their ability to mutate rapidly is what makes them so successful and so dangerous. At the heart of this ability is their genetic material, which can be either DNA or RNA. RNA viruses, like influenza and coronaviruses, are particularly notorious for their high mutation rates. This is because the enzymes that copy their RNA make more mistakes compared to the enzymes that replicate DNA. These mistakes are mutations, and while many of them are harmless or even detrimental to the virus, some give it new abilities, like escaping the immune system or jumping to a new host species. When a virus infects a cell, it hijacks the cell's machinery to make copies of itself. During this replication process, mutations can occur. Sometimes these mutations create a new version of the virus that is better at infecting hosts or evading the immune system. This is natural selection in action. The more successful mutations get passed on, while the less successful ones fade away. Influenza viruses are masters of mutation. Every year, they change just enough to evade the immunity we might have built up from previous infections or vaccinations. This is why you need a flu shot every year. Each season's flu virus is a little different from the last. Viruses can also evolve through a process called recombination. This happens when two viruses infect the same cell and exchange genetic material, creating a new hybrid virus. This can lead to significant changes in the virus's properties. A famous example is the H1N1 swine flu pandemic of 2009, which resulted from the mixing of human, pig, and bird influenza viruses. The impact of these mutations isn't uniform across genders. Men and women have different immune responses, influenced by both genetic and hormonal factors. For instance, testosterone tends to suppress immune response, while estrogen can enhance it. This means that men might be more susceptible to severe infections and complications from certain viruses compared to women. On the flip side, women might experience stronger inflammatory responses, which can sometimes lead to different health issues. Behavioral factors also play a role. Men are more likely to engage in behaviors that increase their risk of exposure, such as smoking and drinking, which can weaken their immune system. Women, meanwhile, are often primary caregivers and may have higher exposure to sick individuals, increasing their risk of infection. Cross-species transmission, how viruses jump hosts. Now let's delve into how viruses jump from one species to another, a process known as cross-species transmission or spillover. This is where viruses get creative, swapping hosts like a college student swaps majors. Cross-species transmission is a key factor in the emergence of new infectious diseases. The process starts when a virus from a natural reservoir, like an animal, comes into contact with a new host species, often humans. This can happen through direct contact with the animal, its bodily fluids, or through intermediate hosts. Intermediate hosts, like pigs or birds, act as a stepping stone, allowing the virus to adapt before it makes the leap to humans. For instance, the H1N1 influenza virus involved a mix of avian, swine, and human influenza viruses. For a virus to successfully jump species, it needs to overcome several biological barriers. First, it has to bind to and enter the cells of the new host. This involves specific interactions between the virus's surface proteins and the host cell's receptors. If the virus can latch on to the host cell's receptors, it can enter and begin to replicate. However, this isn't a given. The host's immune system is a formidable opponent, and many viruses fail at this stage. Once inside, the virus must replicate efficiently and evade the host's immune defenses. Some viruses achieve this by mutating rapidly, constantly changing their genetic makeup to stay one step ahead of the immune system. Others might hijack the host's cellular machinery in ways that suppress immune responses. One infamous example of cross-species transmission is the HIV virus. HIV is believed to have originated in chimpanzees, where it exists as simian immunodeficiency virus, SIV. Through activities like hunting and butchering chimpanzees, humans came into contact with SIV, which eventually adapted to humans and became HIV, leading to the global AIDS pandemic. Another example is the Ebola virus, which is carried by fruit bats. Humans can contract Ebola through contact with infected bats or other animals that have come into contact with bat saliva or feces. This can occur during hunting, handling of bushmeat, or through other means of close contact with infected animals. Gender-specific behaviors and roles can influence the risk of cross-species transmission. For instance, in many cultures, men are more likely to engage in hunting and butchering of wild animals, activities that bring them into close contact with potential viral reservoirs. Women, on the other hand, may be more involved in food preparation and caregiving roles, which can also expose them to zoonotic diseases. In both cases, cultural practices and occupational roles contribute to differing levels of exposure and risk between genders. It's important to note that cross-species transmission is not a one-time event. For a virus to become established in a new host species, it typically requires multiple spillover events and sustained human-to-human transmission. This is why certain practices, like improving hygiene and reducing wildlife trade, are crucial in preventing these events from happening in the first place. Laboratory and synthetic origins. Let's pivot to a topic that's both intriguing and controversial: the role of laboratories in the emergence of new viruses. While most viruses come from natural sources, there are instances where human activities in scientific labs have played a part, either accidentally or intentionally. Accidental releases from laboratories can happen due to breaches in biosecurity measures. Labs that study dangerous pathogens, like those handling Ebola or novel coronaviruses, have strict protocols to prevent any accidental releases. However, mistakes can happen. For instance, in 1977, a strain of H1N1 influenza reappeared after 20 years and is suspected to have originated from a lab accident in China or the Soviet Union. Such incidents remind us that even with rigorous safety measures, the possibility of an accidental release always exists. Then there's the intentional synthesis of viruses. Scientists sometimes create synthetic viruses to study their properties and develop treatments or vaccines. This process involves manipulating the genetic material of a virus to understand how changes in its genome affect its behavior. While this research can lead to significant advancements in medicine, it also raises ethical and safety concerns. The creation of the first synthetic polio virus in 2002 and the reconstruction of the 1918 influenza virus in 2005 are examples of this type of research. Biosecurity is a major concern here. High containment laboratories, often designated as BSL-3 or BSL-4, are equipped with advanced safety features to prevent the escape of pathogens. These labs have airtight seals, advanced air filtration systems, and strict protocols for handling dangerous viruses. Despite these precautions, the potential for human error or equipment failure remains. Historically, the field of virology has been male-dominated, but efforts are being made to encourage more women to pursue careers in this area. Diverse perspectives can enhance scientific research, leading to more comprehensive safety protocols and ethical guidelines. Encouraging gender diversity in labs not only promotes equality, but also improves the quality and safety of research. To address these risks, international regulations and oversight are crucial. Organizations like the World Health Organization, WHO, and national agencies enforce guidelines for conducting research on dangerous pathogens. These regulations aim to ensure that research is conducted safely and ethically, minimizing the risk of accidental or intentional release. Global travel and trade, speeding up spread. Our world is more interconnected than ever, and while that's great for business and tourism, it's also a fast track for viruses to spread. The sheer volume of global travel is staggering. In 2019, before the COVID-19 pandemic, there were over 4.5 billion airline passengers worldwide. This constant movement of people across countries and continents means that an infectious disease can spread from one side of the planet to the other in a matter of hours. Airports and airplanes, with their crowded, enclosed spaces, are perfect environments for viruses to hop from person to person. A single infected traveler can unknowingly carry a virus to new locations, sparking outbreaks far from the original source. Trade also plays a significant role. The global movement of goods involves countless interactions among people from different regions, increasing the chances of virus transmission. Additionally, the transport of animals and animal products can introduce zoonotic diseases to new areas. The 2003 outbreak of SARS was traced back to infected civet cats sold in Chinese markets, which likely contracted the virus from bats. The civet cats were then transported to other regions, facilitating the spread of the virus. Historically, diseases like the Black Death spread through trade routes, such as the Silk Road, carried by fleas on rats aboard merchant ships. Today, the spread is even faster due to modern transportation methods. The Zika virus outbreak, which began in Brazil, spread rapidly to other parts of the Americas, Southeast Asia, and beyond, largely due to infected travelers and the global trade network. Tourism is another major factor. Popular tourist destinations, particularly those with limited healthcare infrastructure, can become hotspots for virus transmission. Visitors can contract diseases abroad and bring them back to their home countries. This was evident in the spread of COVID-19, where initial outbreaks were linked to travelers returning from affected regions. The dense population and diverse origins of tourists create a melting pot for viruses, making it easier for them to spread across borders. Public health measures at borders and transportation hubs are critical in controlling the spread of infectious diseases. Screening travelers for symptoms, imposing quarantines, and restricting travel from affected areas are some strategies used to prevent the spread of viruses. However, these measures need to be implemented swiftly and effectively to be truly effective. Digital tools and data analytics are increasingly being used to track and predict the spread of viruses. By analyzing travel patterns and social networks, health authorities can identify potential hotspots and implement targeted interventions. This approach was used during the COVID-19 pandemic to monitor and control the spread of the virus. Climate change and new viral landscapes. As global temperatures rise and weather patterns shift, the habitats of disease-carrying animals and insects are also changing, bringing new risks and challenges. One of the most direct impacts of climate change is on the distribution of vector-borne diseases. Vectors are organisms, like mosquitoes and ticks, that transmit pathogens between humans or from animals to humans. Warmer temperatures can expand the range of these vectors, allowing diseases like malaria, dengue fever, Zika virus, and West Nile virus to spread into new areas previously too cool for these pathogens to thrive. For instance, the Aedes aegypti mosquito, which carries dengue and Zika viruses, has been moving into higher altitudes and latitudes as global temperatures increase. Climate change also affects the life cycle of these vectors. Warmer conditions can accelerate the breeding and biting rates of mosquitoes, increasing the transmission potential of the viruses they carry. Changes in precipitation patterns, such as increased rainfall or flooding, create more breeding grounds for mosquitoes, leading to higher vector populations. Gender differences are evident in exposure and vulnerability to climate change-related health risks. Women, especially in developing countries, are often more affected by climate change because they tend to have more responsibilities related to water, food, and caregiving. These roles can increase their exposure to disease vectors. For instance, women collecting water from mosquito-infested areas or spending more time near stagnant water sources for household chores are at a higher risk of mosquito bites. Men, on the other hand, might be more exposed through agricultural work or outdoor activities that bring them into contact with vectors. Climate change also disrupts ecosystems and wildlife habitats, forcing animals to migrate and come into closer contact with human populations. This increased interaction heightens the risk of zoonotic spillover events, where viruses jump from animals to humans. For example, as permafrost thaws in the Arctic, previously dormant pathogens could be released, potentially leading to new outbreaks. Another aspect is the impact of extreme weather events, such as hurricanes, floods, and droughts, which are becoming more frequent and severe due to climate change. These events can disrupt public health infrastructure, making it harder to control disease outbreaks. Flooding can lead to the spread of waterborne diseases like cholera, while droughts can force wildlife into urban areas in search of food and water, increasing the likelihood of zoonotic transmission. Urbanization and deforestation, driven in part by climate change, also contribute to the emergence of new viruses. As humans encroach on natural habitats, they disrupt the ecological balance and create opportunities for viruses to jump from wildlife to humans. The construction of roads, expansion of agriculture, and development of new settlements all play a role in this dynamic. Immune system variability. Now let's talk about the body's frontline defense against viruses. The immune system. Our immune system is a complex network of cells, tissues, and organs that work together to defend against harmful invaders like viruses. However, this system varies significantly between individuals, influenced by genetics, age, lifestyle, and notably, gender. Genetically, the immune system is influenced by a variety of genes that dictate how our bodies recognize and respond to pathogens. These genetic differences can make some people more susceptible to certain viruses. For example, variations in the ACE2 receptor gene, which SARS-CoV-2 uses to enter cells, can influence how easily the virus infects individuals. Moreover, some people have genetic mutations that impair their immune response, making them more vulnerable to severe viral infections. Age is another critical factor. Generally, young children and the elderly have weaker immune systems. Children's immune systems are still developing, so they are more susceptible to infections. Conversely, the immune response declines with age, a phenomenon known as immunosenescence. This makes older adults more prone to severe complications from viral infections like the flu and COVID-19. Lifestyle factors such as diet, exercise, sleep, and stress levels also impact the immune system. A balanced diet rich in vitamins and minerals supports immune function, while chronic stress can suppress the immune response, making individuals more susceptible to infections. Regular exercise is known to boost the immune system, whereas sleep deprivation can weaken it. One of the most fascinating aspects of immune system variability is the difference between men and women. Generally, women have stronger immune responses than men. This difference is partly due to sex hormones like estrogen, which boosts the immune response, and testosterone, which can suppress it. Women are also more likely to develop robust antibody responses to infections and vaccinations. However, this heightened immune response also means that women are more prone to autoimmune diseases, where the immune system mistakenly attacks the body's own tissues. During the COVID-19 pandemic, these gender differences were stark. Men were more likely to experience severe symptoms and higher mortality rates compared to women. Research suggests that men's weaker immune response to the virus, combined with higher rates of comorbidities like hypertension and cardiovascular diseases, contributed to this disparity. Additionally, behavioral factors, such as higher rates of smoking and drinking among men, which can weaken the immune system, played a role. Interestingly, pregnancy introduces another layer of complexity. During pregnancy, a woman's immune system undergoes significant changes to tolerate the growing fetus, which can alter her response to infections. Pregnant women are generally more susceptible to severe illness from respiratory viruses like influenza. However, the response can vary depending on the stage of pregnancy.